In this video, we will discuss about the complete paper that is Calreticulin mutation affects its chaperone function and also perturbs the glycoproteome. We have discussed some part of this in our last couple of videos. However, we will quickly cover those part and then cover the remaining parts of this paper in this video. So this is the graphical abstract. So what they compared is that they compared the cells of the healthy donor with the against the patient that has this homogeneous homozygous calreticulum or heterozygous calreticulum problems, or the patients that have this uh, JAK2 signaling path activated, JAK2 signaling pathway activated. And they have also compared the wild type calreticulum with the mutant calreticulum and calreticulum knockout to test their hypothesis that is developed from analyzing this comparison. And in order to do the comparison, they have used this LIPMS method, that is limited proteolysis mass spectrometry. And then what they have find out is that uh, there is a change in the glycoproteins formation and calcium regulated proteins. And they also compared simultaneously, they also compared this homozygous calreticulum and uh, knockout homozygous calreticulum mutation and the calreticulum knockout to see its difference. So let us start with the approach that they have used in this paper, that is LIPMS or limited proteolysis mass spectrometry. So what happens in this case is that the proteins are subjected to double proteases. In the first condition, they are subjected to protease that has broad specificity and in their native condition. So what happens? Suppose this proteins is not in, does not have its proper structure, whereas the condition one is supposed to be suppose condition one is the wild type and condition two is the uh, condition two is the in condition 2 is uh, the cells that does not have a particular gene, or we can say uh, interested condition for the experiment. So what happens in condition 1? Condition 1, the cells acquire the native structure since it is a wild type condition. And in condition 2, because of the modified condition, some of the cells might not acquire their native structure as it is shown here. And when they are subjected to a broad uh, protease, and the arrow shows the cleavage side of this protease, so it will cleave of this uh, differently formed structure, whereas in the case of wild type condition there is no differently formed structure. So, and after this first protease, then the normal process happens, that is denaturing and alkylation of the proteins take place. And after that they are subjected to specific diation specific diation protease, that is trypsin or lysis C, that can cleave at specific sites of the amino acid, that is either arginine or lysine. And in this case, since this area is not cleaved in the first protease condition, we will get a single peptide, whereas this area is cleaved in the first protease conditions. And in the after second protease condition, we will have two different peptide. So when they are subjected to mass spectrometry, we can distinguish this differently. We can distinguish this differently structured protein by comparing the peptides. That is, in case of For example, in case of this wild type condition, we will see only a single peptide, whereas in case of this in in case of this uh, interested condition or uh, condition of interest, whereas in case of the condition of interest or different condition, we can see the different peptides as it is shown here in the yellow. So by analyzing the peptides, we can predict whether the protein has uh, whether there is a structural change in the protein or not. And before going further, let us understand the uh, three kind of condition that is frequently used in the paper. First one is the homozygous gene. So it is the identical gene allele present in the cell system, or we can say if a particular gene has uh, exist at different places in the DNA, then both of them are mutated in a similar form. In this heterozygous gene, the different alleles has different mutation. It can be like one is mutated and another is not mutated also. And in case of this hemizygous gene, is, is that is the condition in which only there is one copy of gene available within the cell system, whereas usually more than one copy is there. So this figure focuses on comparing the uh, proteomics data from that acquired from limited proteolysis mass spectrometry uh, in between the healthy donor and the mutated donor, that is Calreticulum homozygous Calreticulum mutated donor, Calreticulum heterozygous mutated donor, and the donors that have this JAK2 signaling pathway activated. So what they have find out is that uh, homozygous Calreticulum mutated donor has uh, more more number of proteins that are that have structural changes and also the abundance changes, and this gray color represents proteins that have both abundance and structural changes, whereas in case of this heterozygous calreticulum mutation and this JAX2 calreticulum mutation, the changes are smalls, and our both of this heterozygous and JAX2 mutated JAX2 signaling pathway activated condition has comparable amount of proteins that are significantly upper that has that has that shows changes that shows significant changes. And later on they also did the volcano plot to confirm this study. They also did the volcano plot to confirm this thing, and they have find out that the homozygous in case of heterozygous and JAX2 signaling pathway activated JAX2 condition, there are less number of proteins that are significantly that are significantly upregulated or downregulated, whereas in case of homozygous calreticulum homozygous calreticulum condition, the amount of proteins that are significantly upregulated or downregulated are more. And later on they compared these proteins of what are the later on they try to identify what are these proteins that are significantly upregulated or downregulated. So they have identified, categorized them into three categories. One is glycoprotein, another one is calcium regulated protein, and all of the others are considered in the others. They have find out that D panel represents the changes in the abundance. They have found out that most of the glycoproteins are most of the glycoprotein in homozygous Calreticulin condition. There is a abundance change and also the structural changes occurred. Oh, this is abundance. This is the abundance panel. So there is abundance change in the uh, glycoproteins and also most of the proteins that are non-glycosylated. You can say that. There is less number of proteins that are calcium regulated, shows abundance change. And when we see the common proteins uh, that shows relative change in the abundance between homozygous calreticulum condition and heterozygous calreticulum condition, they are very small. This indicates that in homozygous calreticulum condition, the number of proteins that are that are related, the number of, we can say that in homozygous calreticulum condition, uh, there is additional signaling pathway that get dysregulated, or additional changes might occur in this cell lines compared to the heterozygous calreticulum condition. And same thing they did with the structural changes in the protein. And here also they have find out the similar conclusion, that is uh, in case of calreticulum and the common proteins between homozygous calreticulum condition and heterozygous calreticulum condition are small. So the cells in case of homozygous calreticulum condition will show more, has shown, will might have more disruption or more changes compared to the conditions in which compared to the this heterozygous calreticulum condition. And then they did some gene ontology to find out the categories of the proteins that are that show that are that shows relative abundance more than the threshold, and also that shows the structural changes, that shows some significant structural changes. So they have highlighted those conditions in this panel A, and the gray area represents the missing value, that is they don't have the values whether these are significantly uh, whether there is a relative significant changes in the abundance or not. So in this we can see that and this condition is about the homozygous calreticulum versus the cell lines with JAK2 signaling pathway activated. So what they find out is that there is not much change in the relative abundance, whereas there is much change in the uh, stu- structural characteristic or structure of the proteins. And when and this panel B indicates the figure in which the number of the genes that are that shows uh, genes that shows structural changes are represented in the panel B, in which the x-axis represents the number of peptides that are identified uh, to have structural changes compared to the control condition, and this color indicates the a log 2 fold change in the case of this relative abundance. So and this C panel indicates, and the C panels also indicates about the gives information about the gene ontology, that is the proteins that are included in the disulfide bond, uh, out of which around this 60% of this uh, this panel C indicates about the how categorizes the proteins that are significantly that shows significant relative abundance change or the structural change. So in this they have categorizes the disulfide bond. They have categorizes the disulfide bond proteins, in which around 60% of the protein disulfide bond proteins are the glycoprotein that are significantly that show significant changes. And this calcium most of the calcium regulated proteins are either cytoskeleton or actin binding protein, and some of these UBI conjugation proteins are the mixture of glycoproteins and calcium regulated proteins. Later on they did to see if there are any relative changes. Later on they also did also check whether there is a relative abundance changes in the specific category of the proteins or not. And so what they have identified is that the glycoprotein shows a significant changes in the relative abundance, whereas the calcium regulated proteins does not show any or much changes in the relative abundance, and the other proteins also show changes in the relative abundance. So from this they come to a point that most of the glycoproteins, uh, we can say that calreticulin mutation not only affect the structural changes in the glycoprotein, but also the relative abundance of the glycoproteins are also reduced. And the structural changes are change in the structural of the glycoproteins can be seen from this panel, and the relative change in the relative abundance of the glycoprotein can be seen from this panel. And this figure is about the homozygous calreticulin condition. And this panel also talks about the homozygous calreticulin condition. So in this the panel A shows the same thing, that is structurally altered peptide per protein for each proteins, the number of peptides that are altered, and the y axis shows the fraction of peptides that are altered for that particular protein. So in this from this figure the researchers are able to identify that this MPO glycoprotein, this some of the glycoprotein, that is MPO CTSS, shows a more MPO shows a more number of peptides that are altered, and CTS shows a more percentage of the peptides that are altered. And whereas this LCP1, which comes under the calcium regulated proteins, also shows the more number of peptides that are altered. So they pick out some of the proteins from this figure and did the detailed analysis. So first they did with the glycoprotein, that is MPU. So they are able to find out almost all the peptides that can form the complete structure of this MPO protein, and the red color indicates the significant structural changes in the peptide, and gray color indicates the all detected peptides. So in this figure it seems like that the complete structure, or there is a global structural changes of the MPO protein has happened. And same thing they did with the another glycoprotein, that is CTSS. And in this also they find out that most part of the proteins are significantly altered. Then they did the things same things for two calcium regulated proteins. So the problems that they have identified, they face in this is that they are not able to identify uh, they are not no no in this figure, this in the panel D figure, what they have find out is that most of the peptides, significantly structured peptides, they have find out are has one common thing, that is they are either uh, in the EF hand, or they somehow in conjugation with the EF channel, EF hand domain. And same thing they have find out in this another calcium regulated protein, that is S100412, that is at either EF hand 1 or EF hand 2. So from this so far they made a point that in case of glycoprotein there is a global structural changes might have occurred, and in case of calcium regulated protein there is a changes at significant only at localized changes, structural changes might have occurred, that is around this EF hand. And this figure F, or panel F, depicts their hypothesis that they have developed so far, that is uh, here the free structure of the protein is there, and when it is interacting with the calcium there is a change in the structure that is shown here. So and this is the general figure. So when they have uh, used the publicly available figure, that is LCP1N S100A12, and the red peptide or structure that is shown in the red represents the significant structural changes in the peptides that might have occurred both in case of LCP1 and S100A12. And this panel G shows the change in the relative abundance and the uh, significant structural changes of the uh, proteins that belongs to which category. So we can see that there is very less categories of the proteins that shows relative abundance, whereas most of the proteins, most of this category shows structural changes in the abundance for calcium regulated protein. For this for calcium regulated protein, and this thing in which the first one is that of this EF hand domain. And so far this panel is done. So now they move on. So far they have made a point that homozygous calreticulin mutation will affect the glycoprotein, relative abundance of glycoprotein, and also made structural changes, global structural changes in the glycoprotein. So now they wanted to identify the compare between the Calreticulum knockout condition and Calreticulum hemi condition. So they did the same thing. So now they did the same analysis, that is they identify the proteins that are that shows significantly that shows. Now they are comparing the condition between cell lines in which calreticulum is completely knocked out and the condition in which calreticulum is hemizygous calreticulum condition, that is only gene of one gene of calreticulum present, whereas in usually more than one genes are present. So in this they have identified the changes. They have compared the changes, and both the changes are almost comparable. There are not much significant changes in both relative abundance of the protein and structural changes. And then they also did compare the expression of calreticulum in different condition, and in case of this knockout condition it is significantly down regulated, whereas in case of other conditions, that is in hemizygous condition and homozygous conditions, they are down regulated but not to the extent of this calreticulum knockout condition. And then they check whether there is a structural changes in the calreticulum protein or not. uh, and they have identified that number of they have identified that the number of peptides that shows this calreticulum knockout in this panel. And then they also did the abundance changes, also find out the abundance changes of the proteins, whether this abundance changes in the form of the number of peptides that they are able to identify. And these things are comparable, as this one shows the scale of log2 fold change, whereas this one shows in the form of number of peptides. And when they did the gene ontology analysis, they find out that three kinds of proteins are show significant changes, that is glycoprotein disulfide bond and signal proteins. and and then they try to find out what is the common between this hemizygous condition and also this knockout condition. And they have find out that around 20% of the proteins are common both in structural changes that show structural changes and just shows relative abundance change. And the final panel shows uh, the list of chaperones that are changed in this condition. And they find out that most of the chaperones are shows abundance change, and out of which some of them shows structural changes also. So this could be the reason why there is a change in the abundance and structural of these proteins, that is glycoprotein disulfide bond and signal, because chaperones helps the proteins to acquire the native structure. And these proteins in the in insufficient amount of chaperones or infunctional chaperones might not be able to get their native structure. So now they decided to identify the function of the PDI, it is protein disulfide isomerase. For this, they choose the candidate that is MPO, which they have identified from this figure, and also they have the information more almost maximum information of these proteins from the limited proteolysis mass spectrometry. So this is the actual process that they have shown how this MPO protein is formed. It interacts with calreticulum, then calnexin, then the dimer is formed, and then it is uh, secreted into the from end up that it is secreted uh, in the cells or from the cells for its function. So panel B shows the flow cytometry figure in which they have checked the MPO protein in the calreticulum wild type condition, in the calreticulum knockout condition, and this dashed line indicates the unstained condition. And this thing they have done in the HL60 cell, and another thing that they check this flow cytometry condition in the case of this calreticulum homozygous in the patients that have this PMN or this myeloproliferative neoplasm condition, in which the homozygous calreticulum condition exists. So far they got almost the similar amount of MPO. Then this is the MPO expression uh, in relation to the control, in which the control condition is considered as the 100%. So what they have identified is that in case of this PMN condition, the patient 4, patient 9, or we can say this calreticulum homozygous calreticulum expression of this MPO in homozygous calreticulum and this calreticulum knockout is low compared to the heterozygous calreticulum and JUG2 mutation. Whereas in case of HL60 cells, the two types of clones that they have used in which the calreticulum is knocked out. So in both the conditions, the MPO expression is less. Then they need to check whether they need to check whether the interaction of this MPO with calreticulum and calnexin. So for this they have used the reagent BFA. So what this BFA does is BFA will inhibit or will prevent the transfer of proteins from endoplasmic reticulum to Golgi. So first they did it without BFA. So in this they have used wild type, and this T1 T2 T1 is the calreticulum mutated condition 1 and calreticulum mutated condition 2, and this is calreticulum knockout. So in the wild type they are able to find out this MPO protein, and in case of this calreticulum knockout condition they are not able to find out that much MPO. And when they check this mutated calreticulum, they can see that the calreticulum is not there in other condition except T2. In T2 some of the calreticulum is still exists, mutated condition, whereas And when they did the Western blot also for the actual calreticulum, and for this T1 and T2 they use the antibodies to detect this calreticulum mutated condition, in which they use the antibodies that can detect the mutated C terminal of this calreticulum. So without BFA they are not able to find out, because one reason that they mention is that calreticulum might not retain within the cells or degrade itself, uh, because of the absence of the KDL sequence. And when they did the same experiment with BFA, and they are able to see the mutated calreticulum that are available. However, there is no much interaction, no much change observed in the MPO, and they use the control to check whether the MPO is there in the wild type condition or not. And in the HL60 condition, they have used two cell system, HL60 and the MAR cells. So they can see the MPO heavy chain and apopro MPO in the controls and also in the wild type. Whereas in case of this mutated condition, they cannot see the MPO. So from this, they made a point that the mutated calreticulum might not interact well with this MPO. So because of this, the MPO will not be able to form a stable structure. So and then they did another quantification to check the effect of this BFA. That is, in the presence of BFA, how much this calreticulum is increased. In case of wild type, there is not much increased, as calreticulum is already has the signature protein signature sequence to stay in the endoplasmic reticulum. Whereas in case of this mutated 1 and mutated 2 condition, there is an increase in the calreticulum, uh, and in the case of knockout condition, there is uh, only down regulation of the calreticulum has observed. And they did the similar experiment, but instead of using BFA, right now this time they used or they modified the sequence with KDEL. That is, KDEL is the sequence of amino acid, uh, that are sequence of protein. Is the sequence of amino acid that are present in the proteins who are who are that are present in the proteins whose final destination is ER lumen. This all ER lumen proteins will have this KDEL sequence. So when they did it with the wild type, the there is a MPO protein is there, and calreticulum is also there. And next they did with wild type by removing this KDL sequence. So in this case, they are able to see the calreticulum. However, the MPO interaction got reduced. And then they did the T1 and T2 as it is, and they were able to see small amount of calreticulum, mutated calreticulum. And next they did is they introduced this or modify the calreticulum mutation with KDEL sequence, and then they are able to see the calreticulum mutation. However, there is no interaction with the MPO. And here they have used the control, that is empty vector, to see it there. is the impact of vector or not. And this empty vector with MPO, and there also they're not able to see anything. And then they use the untransfected proteins, then they also not able to see anything. And this experiment is done on the knockout cells, calreticulum knockout cells. uh, so and then they quantified the MPO expression in the similar condition, and they can see that there is significant decrease in the calreticulum, a significant decrease in the MPO expression when the KDL sequence is removed from the wild type, whereas there is not much change in the other condition, as the MPO is not interacting with the knockout condition. Whereas when the wild type calreticulum is modified, its interaction with MPO will also get affected. So this panel indicates that the glycoprotein, that the MPO glycoprotein interaction with the mutated calreticulum is reduced, or the calreticulum's chaperone functionality is reduced in case of mutated condition. Next, the final panel that they used to check whether it also affect the mutated calreticulum also affect its functionality on this disulfide isomerase, protein disulfide isomerase, or we can say the proteins from PDI family, more specifically ERP57. So for this they used the same strategy as they did for this MPO to identify MPO proteins, and they have find out that this time they check the calreticulum amino acid sequence, and they have find out that there is a significant changes in the at this one some of the significant changes in the structure occurred in the end domain site, and some of the significant changes occurred in the start of this P domain site. And when they check this structure, and they have seen that the P domain sites, that this structural changes will interact with the protein isomerase family, or we can say ERP57, that helps the ERP57 and N-glycan precursor to come in close contact with each other, so that ERP57 can helps the proteins to get their native structure. So, and this there is a change in the peptide belongs to that region, as it is shown in the red color. So from this there is a motivation, or there is a positive sign that comes that uh, there is a structural changes in the calreticulum that might cause or affect the interaction of this ERP57. So in order to identify this, what they did is uh, they use this uh, PLA assay, or we can say proximity uh, ligation assay, to detect whether there is a effect of this mutated calreticulum on this ERP57 interaction or not. So this is the control experiment. They use different antibodies to detect this calreticulum, and also the protein, MPO protein, calreticulum and the MPO protein. This is the control experiment. It does not have any MPO protein. It does not have any calreticulum. And this is the calreticulum knockout condition. And this is the calreticulum mutated condition without BFA. Mutated T1 condition, and this is the calreticulum mutated T2 condition without BFA. And this is the calreticulum mutated condition with BFA, and this is the calreticulum mutated 2 condition with BFA. So in this figure we can see that the interaction of this calreticulum mutation is low in case of without BFA condition, and is increased with case of BFA condition. So this gives a indication that positive signs that calreticulum might interfere, or might mutation in the calreticulum might affect the interaction of ERP57. As if there is no change in this, then we could have our hypothesis could if there is no changes in these two these two figures, these first two and the last two figures, then we could have made a point. We could have think that there is not much change in the ERP57. However, there is some changes that we can see. Then later on we quantify this thing, and we are able to see that the signal of this calreticulum mutation and MPO is increased in case of this mutated condition, and they are increased more in case of this mutated condition with BFA. And when they compared, this is the control experiment, that is calreticulum knockout and this calreticulum wild type, they also see that the signals are positive only when the calreticulum wild type is present. Later on, since this experiment is not designed in a way that they can compare the wild type and mutated form directly, so they have used another assay. Researchers have used another assay to compare the calreticulum wild type and MPO interaction and calreticulum mutated and MPO interaction. First, they modify the calreticulum with GST, and then they are able to measure the abundance at absorbance at 450 nanometer, and they can see that the difference between wild type and the mutated condition. In case of wild type, the signals are quite high compared to the case of this calreticulum mutation. And then since the protein structure is not available for MPO, so they have used the MHC protein structure, and they have identified the peptides that show significantly structural changes in the form of red spot, and they can see that the region that is responsible for the interaction of this glycan calreticulum and this ERP57 is close to or we can say the peptides that show significant structural changes close to the region where there is an interaction of this ERP57 proteins, glycan and calreticulum. So this hypothesis uh, might looks promising that this changes in the peptide structural might affect the interaction of this ERP57 with this calreticulum or this peptide or this protein. So later on they did the similar study with this calreticulum mutation with ERP57, and they can see that in case of this calreticulum knockout and mutated condition, there is a down regulation of this ER57 occurred, whereas in case of this knockout and other mutated condition, there is not much significant changes. And when they compared this control experiment, this calreticulum knockout with wild type, they can see that the signals are quite high in case of this wild type. So from this, they made their point more. They, this experiment will also support their previous point that ERP57 interaction is more in case of wild type calreticulum, whereas in case of knockout or mutated condition, interactions are less. And this thing is presented here in the form of this protein diagram or protein tertiary structure, protein cartoon structure, in which this significantly structural peptide, we can see the significantly structural peptide that are highlighted in red is close to the proximity where the glycans interact with calreticulum and this ERP57. So what is the summary, what is the conclusion from this paper is that calreticulum mutations leads to quantitative loss of functional trait. I made a mistake. Not just quantitative loss but also the qualitative loss of this functional trait. The that exhibits proteins that exhibits protein exhibits structural changes in the cytoskeletal and calcium related protein, but the abundance change of these proteins are not much affected. Whereas in case of glycoproteins, in case of this homozygous calreticulum mutation, both the structural and expression of the glycoproteins, not the proteins, not just the proteins, the glycoproteins get affected. So this paper provides a more insight about how this calreticulum mutation affects the glycoprotein and these other proteins, that is cytoskeletal or calcium regulatory proteins. If you need to understand this thing details or clear any of your doubt, you can either write a comment or go through the reference that has been provided in the description. That's all for this video. Thank you for your time.